Dear students, we were discussing about the different methods of birth control. In the last class, we have studied about natural methods of birth control, and today we are going to study about artificial methods of birth control. There are different artificial methods which includes barrier methods, then application of spermicides, intrauterine devices, hormonal methods, surgical sterilization, and medical termination of pregnancy. Let us see all these methods one by one. First is barrier method. Barrier method prevents the sperms from entering into the uterus, and you know that the sperm is deposited in the vagina of the female during sexual intercourse, and the sperm travels up the uterus. It enters in from the vagina, it enters into the uterus through the cervix, and it moves up the uterus, and in the fallopian tube fertilization occurs normally. And if the entry of sperm into the uterus can be prevented, fertilization and subsequent pregnancy can be prevented. So barrier methods prevent the entry of sperm into the uterus. There are different barrier methods which are used, that includes condoms, diaphragm, and vaginal sponge. Condoms, they are thin latex rubber sheets, and there are two types of condoms which can be used by male as well as the female. There are male condoms and female condoms. Male condoms, they fit on the erect penis, and the sperms which are released during the ejaculation process will be collected in the male condom. They will be trapped in the condom, and the sperm is not deposited in the vagina of the female. So fertilization can be prevented. Then on the right side of this picture, you can see the female condom. The female condom, it is also a thin latex rubber sheath. It is actually fitted into the vagina of the female during sexual intercourse. So it is having two ends. One end is open and the other end is closed, and it is fixed into the vagina of the female during sexual intercourse. And here you can see this end is closed, and the sperm will not be entering into the uterus. That is how fertilization is prevented. So these are the condoms. Then diaphragm, it is also a barrier method. Diaphragm is a rubber plastic cup that is fitted onto the cervix of the uterus. It is inserted into the vagina, and here it is the cervix of the uterus, and this is the uterus. So the diaphragm is fitted on top of the cervix, so the entry of sperm into the uterus is prevented. Sometimes the diaphragm will also contain certain spermicidal substances which can kill the sperm that is deposited in the vagina. Then vaginal sponge, it is actually a barrier method in which a small absorbent piece of sponge which is impregnated with some spermicidal substance, it is inserted into the vagina, and during sexual intercourse the sperm that is deposited in the vagina will be killed by the spermicidal substances that are present in this vaginal sponge. Then application of spermicides. Spermicides contain a sperm killing agent which is nonoxynol 9, and it can also contain certain acidic substances like lactic acid, citric acid, boric acid, potassium permanganate, and zinc sulfate. And these are available in the form of foam tablets, jellies, pastes, or creams, and these spermicidal substances are applied into the vagina during sexual intercourse, and the sperms will be killed by these spermicidal substances. Then intrauterine devices. Intrauterine devices are small devices which are inserted into the uterus, and they are fixed inside the uterus. Here in this picture you can see they are fixed inside the uterus, and the presence of these intrauterine devices will prevent the will prevent the implantation of the embryo in the uterus. The sperm will be deposited in the vagina during sexual intercourse. The sperm is entering into the uterus through the cervix, and the fertilization occurs in the fallopian tube. Here you can see the ovary. Then this is the fallopian tube. Ovum will be released from the ovary, and the sperm and the ovum, they unite together to form the zygote in the fallopian tube, and this zygote will develop into the embryo, and the embryo is getting attached to the wall of the uterus, and development occurs. This is the normal case that occurs if fertilization occurs. And the presence of intrauterine devices will prevent the attachment of the embryo to the wall of the uterus, and the embryo will be discarded outside. And that is how fertilization is prevented. Then there are three types of intrauterine devices. Non-medicated devices, they are the first generation intrauterine devices. And the second generation intrauterine devices includes the copper intrauterine devices, or the copper T. It is called copper T. In this picture you can see the copper T. The copper T is actually a plastic material, and the copper wire is wrapped around the stem. Here you can see the copper wire is wrapped around the stem, and the copper ions will slowly dissolve in the uterine fluid, and it will be dispersed in the uterine fluid, and the presence of this copper ions will prevent the implantation of the embryo, because it is changing the ionic environment or the chemical environment inside the uterus, and it will prevent the implantation or the attachment of the embryo onto the wall of the uterus. So the development cannot occur and pregnancy will not occur. Then the third generation intrauterine devices are hormone releasing intrauterine devices. Here you can see the hormone releasing intrauterine devices. They will release small amounts of hormones into the uterine fluid, which will prevent the implantation or the attachment of the embryo onto the wall of the uterus. Then hormonal methods. Hormonal methods include birth control pills or the oral contraceptives, morning after pill, hormonal implant, and contraceptive patch. Here you can see the oral contraceptive. These are tablets which are regularly taken by a female to prevent the cyclic changes which are taking place in the ovary. So the ovum, development of ovum in the ovary will not take place, and ovum will not be released, and in the absence of the ovum, even if sexual intercourse occurs, pregnancy will not occur. Then this is a mixture of synthetic estrogen and progesterone, and in the body of the female normally estrogen and progesterone will be produced, but through this oral contraceptive extra amounts of estrogen and progesterone are taken in, and the presence of this estrogen and progesterone in the blood will prevent the secretion of pituitary and hypothalamic hormones, and this deficiency of pituitary and hypothalamic hormones will prevent the development of ovum in the ovary, and also ovulation is also prevented, and that is how pregnancy is prevented in the case of oral contraceptive. Then morning after pill, it is taken after an unprotected sexual intercourse. If a female forgets to take the regular oral contraceptive, and if she happens to have an unprotected sexual intercourse, a morning after pill can be taken, and this contains synthetic progesterone like hormone, and this prevents the sperms reaching the ovum, or it can prevent the implantation of the embryo. Then this is the hormonal implant. It is actually an implant of the size of a matchstick, and it is inserted under the skin. It is inserted under the skin, and the hormonal implant will release small amounts of constant amounts of hormone into the blood, and it will be synthetic estrogen and progesterone, and that will do the same function as in the case of oral contraceptive. It will prevent the release of hypothalamic and pituitary hormones, and the cyclic changes taking place in the ovary, and the development and release of ovum will be prevented. So that is hormonal implant. Then contraceptive patch. Contraceptive patch, it is actually a transdermal patch which is applied on the skin of the woman. And as in the case of hormonal implant, this transdermal patch or the contraceptive patch will release small amounts of synthetic hormone into the body of the female, and the cyclic changes in the ovary is prevented. That is how this is functioning. Then surgical sterilization, that includes tubectomy, hysteroscopic sterilization, vasectomy and vas occlusion. This is tubectomy, or it is otherwise called tubal ligation. Normally, the sperm is deposited in the vagina and it is entering into the uterus, and it is meeting with the ovum in this fallopian tube, and fertilization occurs, and the embryo will travel back to the uterus and it will attach to the wall of the uterus and development occurs. This is normally occurring. But here in this case, the fallopian tube is cut and tied. So the ovum will not the ovum that is released from the ovary will not reach here, then the sperm will not meet the ovum. So the meeting of the sperm and the ovum is prevented because the fallopian tubes are cut and they are tied together. So that is tubectomy. Then hysteroscopic sterilization. In the case of hysteroscopic sterilization, a hysteroscope is inserted through the vagina of the female, and a coil which is called a micro insert or the essure, it is placed in the fallopian tube. So the fallopian tube is blocked and the meeting of the sperm and the ovum are prevented. And this is hysteroscopic sterilization. Then vasectomy, it is done for males. Hysteroscopic sterilization and tubectomy are done for females, whereas vasectomy and the vas occlusion, these two techniques are done for the male. In the case of vasectomy, here you can see the testes which are placed inside the scrotum. A small incision is made on the scrotum, and vas deferens, this duct vas deferens will be exposed. Vas deferens is the duct that is carrying the sperms which are formed in the testes, and the sperms are carried outside during ejaculation through this vas deferens. So the vas deferens is cut and the ends are tied. So the release of sperm from the testis is prevented. The sperms will not be released during ejaculation. So this semen that is released during ejaculation will not contain sperms. So that is vasectomy. Then vas occlusion. Here in this case, a silicone gel is inserted into the vas deferens. Here the vas deferens is not cut. Instead of that, a silicone gel is placed inside the vas deferens, and it is blocked. So the vas deferens is blocked, so the sperms will not come outside. In the case of induced abortion or medical termination of pregnancy, intentionally the fetus is expelled outside. And in India, Medical Termination of Pregnancy Act was enacted in 1971 and it was amended in 2002. According to this law, MTP is legally permitted only up to 20 weeks of gestation, and abortion is not morally acceptable to all people. In the first 12 weeks of pregnancy, abortion can be performed surgically by vacuum aspiration. That is, the cervix of the female is dilated and a vacuum aspirator is applied, so all the contents of the uterus, they are drawn outside through the aspirator tube. And after 12 weeks of pregnancy up to 16 weeks, that is from 13 to 16 weeks, physical scrapping is done to induce abortion. Then in that case, a catheter is inserted through the vagina and the fetus is cut into pieces, and all the contents of the uterus, they are physically scrapped off and their contents are drawn outside. Then after 16 weeks of gestation, abortion is induced by injecting urea solution into the uterine cavity or by administering oxytocin hormone to the female. So the walls of the uterus will contract and the fetus will be expelled outside. So these are the techniques for medical termination of pregnancy or abortion. So that is about artificial methods of birth control. Thank you.